Okay, so we can pick up where we left off on Tuesday. We had talked about all of the prokaryotic and eukaryotic organisms that we sometimes classify under this umbrella of microbes. And just to kind of orient you in terms of the overall tree of life, where we're looking at things, we take bacteria and archaea and see them over here. They're their own separate branches. So even though they share that huge morphological characteristic of not having a nucleus, they are their own separate lineages. They are not really that closely related. And then our third big branch on that tree of life are the eukaryotes, right, that have a nucleus. And what you'll notice is that the archaea are right there in the middle. And what a lot of genetic and molecular analysis has shown us is that in many ways, archaea are more closely related to eukaryotes than they are to bacteria. So just because they look alike doesn't mean they are. And we'll talk a lot more about that as the semester goes on. So bacteria and archaea are their own two domains. Eukaryotes are the third domain, which will have all of our eukaryotic organisms. And again, we'll be talking about some of the protists, some of the fungi, and some of the animals in this class. To give a brief history of microbiology, microbes are theorized to be the first things on the planet, the first living things. Believe it or not, there are fossil records that demonstrate that microbes were present before eukaryotes and before multicellular organisms. So there is a lot of evidence that supports microbes being the first living things. And by our best estimates, scientists believe that cellular life began about 4 billion years ago, probably give or take. None of us were there, we can't say for sure. Best estimates. We estimate that it then took a while before we finally had organisms that produced oxygen. So prior to this, all organisms did respiration in anaerobic conditions. There wasn't molecular oxygen available for them. Not that oxygen didn't exist, it just didn't exist in the form that we know of it today. It could be found in carbohydrates, it could be found as parts of other macromolecules, but it wasn't air that you breathe. And it wasn't until the evolution of the cyanobacteria, which are photosynthetic bacteria, but they're called oxygenic photosynthetic bacteria. They do photosynthesis and produce oxygen. There are microbes that do photosynthesis. They use light energy to drive carbon fixation, but do not produce oxygen as a byproduct. So there is anoxygenic photosynthesis, but this was the first oxygenic photosynthesis. And the idea that there was oxygen available meant that organisms that evolved to be able to not only tolerate oxygen, but to be able to use it, had a whole surface to explore. What we tend to see in a lot of organisms that are anaerobic, especially in bacteria that are anaerobic, they don't require oxygen, is that not only do they not require it, but it kills them. Oxygen generates reactive oxygen species that can damage DNA and proteins. It does that in us too. But we make, and some bacteria make, enzymes to detoxify reactive oxygen species. Many anaerobic bacteria do not. So it's not that they just don't use it. If they are a true anaerobe, it will kill them. Not as quickly as, oh my gosh, they were exposed to oxygen, they're going to die. But relatively quickly. So organisms that evolved mechanisms to detoxify oxygen and then evolved mechanisms to actually utilize oxygen for respiration ended up being a little bit more successful. Aerobic respiration, using oxygen as the final electron acceptor at the end of the electron transport chain, generates more ATP than using something else. That's another big misconception. A lot of people think that anaerobic respiration is the same thing as fermentation, where you only get a couple of ATP. True anaerobic respiration, glycolysis, Krebs cycle, electron transport chain, the same as aerobic. The difference is at the very end, anaerobic respiration can use something like hydrogen or hydrogen sulfide or nitrate. It uses something else as the electron acceptor. But if you get deep into the chemistry of all the redox reactions, which is beyond the scope of this class, what you find is that oxygen, just molecular oxygen, has the best pull for electrons. So you just get the best pull down the electron transport chain. You get the most hydrogens pumped out. You get the most ATP generated. It's the most efficient. So organisms that could exploit that and that could tolerate it flourished, especially on this whole new area of the surface. They were able to live on the surface. Geologists can even point out when this happened, between 2.5 and 3.5 billion years ago, because you can actually see the formation of things called, they're like banded iron formations. You can see when the oxygen in the atmosphere started to react with metals on the surface. So it's not just, fossil record, it's actually looking at the rocks and analyzing the geochemistry there that let us know that now there's oxygen in the atmosphere where there wasn't before. Humans began using microbes, even though they didn't know what they were, at least in documented ways, around 8,000 years ago. These are some of the first recorded uses of microbes. And we know they used microbes because they made fermented products like beer, vinegar, and wine. They didn't know why it worked that way, right? Because they didn't know that microbes existed. They didn't know that cells existed. But they knew that if they, used fermented products, that they could have things like beer and wine and vinegar. Ancient cultures also knew that diseases could be transmitted from person to person. They didn't know how, right? They didn't know that there could be like viruses floating around or soil bacteria that get in a wound, but they knew that people could get sick. Sometimes they had silly ideas about what caused it. Malaria, right? Bad air, which we now know is caused by a parasite. It's transmitted by mosquitoes, but they thought things that were stinky could make you sick. They thought, if you were poor, you were going to get sick, because only poor people get sick. If you're a sinner, sinners get sick. If you're not the same racial background as the dominant culture, you get sick because you're not the right race. They had some silly ideas. But they did know that diseases could be transmitted. And they also knew that in a lot of cases, if you ever got sick once with something, you were very unlikely to get sick with it again. So they documented things like immunity. So they had some silly ideas, but they also knew some things. But of course, there was a major breakthrough that allowed people to figure out that there were microbes. Anybody know what that was?
The microscope?
Yes. Yeah. Probably we're using that today in the lab. If those of you have the morning lab where you're using the microscope, yep. Afternoon lab, sorry, spoiler alert. You're going to use the microscope. So the first microbes were observed in the late 1600s because that's when microscopes were invented. You can't see something that's microscopic unless you have a microscope. So the first actual observation of cells was a little bit earlier. It was in 1665. A British scientist, Robert Hooke, invented a microscope. And he looked at things like cork, like sections of cork under the microscope, and he said, oh, like look at these really like ordered shapes and these ordered structures. He called them cells. And he called them cells because to him they resembled the rooms of a monastery, which were called cells, like a prison cell, except for monks, who are not in prison but live in the monastery. So that's why they're called cells. That's why it's the same word. But again, he was looking at mostly plants because we know that animals don't have that rigid, rectangular repeating structure of cells like that. So he was looking at plant sections. And, those observations eventually led to what we'll call the cell theory. What's more interesting for our purposes as people in a microbiology class was the development of the microscope and the observations of actual microbes. So the first documented, and likely the first, observations of actual living microbes was by a Dutch, I bet you're thinking I was going to say scientist, Dutch tailor named Antony van Leeuwenhoek. He invented his own type of microscope because he wanted to see threads better. He was an actual tailor. And then he was like, well, I can see my threads pretty well. What else can I look at? So he looked at everything, literally everything. He looked at his feces. He looked at his saliva. He looked at everything. And when he looked at these samples, he noticed that there were tiny living, moving things inside of them. And back then, if it moved, it was an animal. If it didn't move, it was a plant. So he called them animalcules. They were little animals because they moved. And he said that there were more of them living in his mouth than in people in all of the Netherlands, which is accurate. So he wrote letters to the Royal Society of London. And 200 years later, then we knew about microbes. You would think, well, this was 1673, so we knew about microbes. Well, one, he wasn't a scientist. And two, he didn't speak English. Nobody cared about a Dutch letter from a tailor. So it wasn't until a couple hundred years later that his contributions were finally recognized. But you see some of his drawings, and he was right. There were more living things in his mouth than in people in all of the Netherlands. There were more living things in his mouth than people all over the world. So, and to this day, really the language, good or bad, of science is English. And when he didn't speak English and he wasn't a scientist, his work wasn't that important. But eventually, people started to recognize that, yeah, there are living things that are small. And we should think about, well, where do these living things come from? And now we're all looking at that going, well, obviously a living thing comes from another living thing. A cell divides, makes another cell. An animal reproduces, it makes another animal. It wasn't as obvious a few hundred years ago. And so there were two competing hypotheses, because these things both can't be true at the same time. One hypothesis was spontaneous generation, that living things could just emerge from nothing, and that there was some vital force that would form life. The other hypothesis was biogenesis, life begets life. You have to have pre-existing life in order to get life. And again, I know spontaneous generation, we might think, well, that doesn't really make sense, but you also think about some of the most preeminent scientific minds of the time were people like priests, who were also deeply religious, very, very strong, generally men of faith. And literally, like, the first story in the Bible is that God created everything from nothing. So why wouldn't that make sense? God still creates things from nothing. So those are our two hypotheses. So what do you do if you're a scientist and you would like to, as much as possible, demonstrate that your hypothesis is best? You do science. That's what we do. So you have a hypothesis. You have to design experiments to test it. Most of these experiments were designed to support biogenesis. It's sort of hard to disprove spontaneous generation, or to prove spontaneous generation, right? There's a really classic example in evolution where I think it was Bill Nye was challenged to a debate about evolution by a creationist. His name escapes me at the moment. And Bill Nye was like, well, yeah, I'm happy to debate you, and I'd even be willing to change my mind about not creationism, about belief in evolution, if you provide enough evidence. If I provide you enough evidence about evolution, would you be willing to change your mind? No, I'm not willing to change my mind. So when you're doing any sort of scientific experiment, you have to have that willingness to be able to change your mind. It's okay to like your hypothesis. I love my hypothesis. This is my hypothesis. I designed my experiments for my hypothesis. It's okay if your hypothesis isn't true. That's real science. But when there are things that are, it's almost impossible to prove, that's maybe not a good scientific question. So if we think of, my question is, does life arise from other life? That's a decent question. And I can disprove that it spontaneously arose. So the first experiment was done in the late 1600s by an Italian, Francisco Redi. And so his experiment involved rotting meat. Not my idea of a fun experiment, but it was an experiment nonetheless. So his experiment, he had 3 flasks, each with rotten meat. One flask was completely open. One flask was completely sealed. The other was covered with like cheesecloth or gauze. So of course, what he observed after a few days in the completely open jar was the presence of maggots on the meat. And again, you have to remember that these are people who are coming from a perspective of, like, this is all brand new. We are like, yes, flies lay eggs that are turn into the larval maggots on rotten food, obviously. But they didn't know that. They didn't sit and observe the whole process. They knew that if something was rotten, there were maggots. And as expected, there were maggots on the rotten meat. On the sealed, there were no maggots. And on the cheesecloth, there were maggots on top of the cheesecloth, but not on the meat. So again, looking back with 300 plus years of perspective, flies are able to access the meat in the open container. Flies can't smell the rotten meat in the sealed container. Flies can smell the meat through the cheesecloth, but they can't access the meat, so they lay their eggs on the cheesecloth. So then, where did the maggots come from? Again, if we were thinking about spontaneous generation, well, the maggots come from the meat. Rotten things grow maggots. But rotten things did not grow maggots. Something had to come in here and make maggots happen. So this was something that, again, in science, we never prove that a hypothesis is true. We don't prove anything. We support. So this is evidence that supported the idea of biogenesis. So then we did more experiments. By we, obviously none of us were around, but scientists back in the day. An English scientist who was also a Catholic priest boiled nutrient broth. So remember, when you boil, that should kill most things. Boiling is pretty good at killing. And then he put a cork on them, so no air or anything could get in, no organisms could get in. But he saw growth. So where did the microbes come from? Well, again, if your hypothesis is spontaneous generation, you're like, well, he killed anything that could have been in there, sealed it up, prevented anything from getting in, and look, life, it came from nothing. And if you're saying, well, life begets life, and you're like, well, he killed everything and he sealed it up, and you're like, oh, where did the living things come from? So this one was a little, wrench in the plans here, because it didn't really support biogenesis. But maybe there was a flaw in the experiment. I obviously am not going back in time to figure it out. Perhaps, what if he didn't boil it long enough? What if the flask wasn't well sealed? What if the cork wasn't totally, blocking out the air? So the biogenesis people were not deterred. It was a little setback, but they were going to keep going. So we had another Italian who kept going. He did a very similar experiment, boiled some gravy, left the flask open, tons of microbes, as you might expect that. Gravy boiled, he completely sealed the flask this time. Gravy free of microorganisms. So this is good for those biogenesis folks. Life didn't come from nowhere. So what were the conclusions? Right? Well, the biogenesis people were really happy. Like, yeah, this supports our hypothesis. But the spontaneous generation people were like, not so quick. We don't believe you. Can you think of why? What's wrong? Where's the flaw here? What would happen if I put you in a room and sealed it and didn't let any air in? Yeah.
They think that the microorganisms died.
Because he completely sealed it, that no air could get in, he killed all the microbes that way. Anyone that would have tried to live, it would have died because he choked them out. It's a legitimate flaw. And again, it's like, seriously, y'all are debating over like gravy growing microbes, but they were. So his conclusions were dismissed. by folks. So, I mean, this is taking place literally over like 200 years. So in 1858, scientists finally like laid it all out in this is like the theory that we're going to go with. Like he gave it a name, he put it on paper, and he says, yeah, like through all these experiments, you know, this is the one that the scientists are going to get behind. Cells come from a pre-existing cell, biogenesis. And that is the third principle of what we now call the cell theory. So cell theory, every living thing is composed of one or more cells. So in order to be considered living, based on this definition, you have to have a cell, which is why viruses, by this definition, are not living. They don't have a cell. You don't have a cell, you don't get to be living. Cells are the basic unit of life. You can't get any smaller and have it be living. If you pull out a ribosome, it is not a living thing. It is a component of a cell. Kind of the idea of if you took a couch out of your house and put it on the street, you wouldn't be like, look, my house. You know, well, that's the couch. A house it does not make. So a ribosome is not a cell. It's not alive. And then that third, this is the sticking point. Cells only come from pre-existing cells. So a lot of scientists really supported this, but there still was debate until our best microbiology friend, Louis Pasteur, of pasteurization fame. He is considered to be the father of modern microbiology. And again, like we're 200 years out here. He demonstrated that microbes are present in the air and can contaminate solutions. So he disproved spontaneous generation using a really clever experimental design. And that's what we'll see on the next slide. So, I don't know, these people and their broth. Obsessed with broth and gravy and boiling. So they boiled some broth. Okay. Obviously there are microbes present in broth, but once boiled, the broth has no microbes. Okay, cool. So then he heated the neck of the flask into an S shape. He didn't seal it. He put it in an S shape. And what he found was that after the broth had cooled, right? If it's too hot, nothing's going to grow. You got to let it cool. After the broth had cooled, it still had no growth. So this was really close to the previous experiment with boiling in the flask and sealing the flask. But again, that idea of, well, you sealed the flask, you choked him out, you didn't give him any oxygen. This is not sealed. It is open. There is air. But the S-shaped design trapped microorganisms and still allowed oxygen to get through. So this was the one that, you know, anybody who was convincible was convinced. There's always somebody who's not convincible. Okay. And so here's somebody who was not convincible. Or this person was convincible, but he was trying to get rid of those naysayers. So what other people were doing was basically trying to repeat his experiment. And that's good in science. If you publish something, especially something that's that earth-shattering, other people should try to repeat your experiment and get the same results. That just supports your results even more. So they were doing the same thing he was doing, and they were getting different results. They were getting growth. So was Pasteur engaged in funny business? Right? Did he doctor his results in some way? Did he lie? Obviously, it wasn't like an AI video, but, right? People lie. He could have lied. So what John Tyndall did was he realized that the labs that couldn't repeat the results were using different kinds of broth. It's always the broth. They were using what's called hay infusion, which literally has grasses in it. And he hypothesized at this point that there was some bacterial form or some life form within the grasses that was resistant to boiling. And these spontaneous generation people are back here going, sure. Now you make up a whole new life form because you don't like your results. And the same year, a German botanist discovered that there is a form of bacteria that is completely resistant to boiling. He discovered endospores. So boiling is not the same as sterilizing. If there is an endospore, even just one, and endospores are very common in soil bacteria, it will resist the boiling. And when the temperature gets back to its preferred temperature, the endospore will germinate and replicate. So the reason, and then this is, we hate to tell you this, but it's true, a lot of science is luck. So the reason Pasteur's experiment worked is because he was using beef broth, which didn't have endospores in it. If he was using hay infusion, it wouldn't have worked. So now we know endospores exist. They are resistant to boiling, and that's why we have an autoclave to destroy endospores. Okay, questions to this point? Yes?
Do you know how exactly the S-shaped stalk microbes from entering flasks?
Probably just because the movement, like microbes don't move in the air, they move because of the air currents. And so the current probably just wasn't enough to bring them in. There just wasn't enough air, like, fluctuation.
Because oxygen was able to enter the S, but...
I think there was probably just enough oxygen in there, not that it entered, but, like, you know, there's just so much oxygen in there that, yeah. The respiratory processes would eventually, like, if there was living things in there, it would eventually start to pull, and that could bring microbes in. But a lot of airborne contaminants are not bacteria. Most bacteria don't float very well. They're fungal spores, which are even bigger. And so they're harder to get to move, but when you get like the AC going or a fan going, they'll blow everywhere. Yeah.
How did they know to boil it?
Because that's just how they knew to like make their food safe. So they knew that like heating would, you know, make food safe. Yeah. Get rid of growth. It's like they knew that keeping things cool would also prevent them from like going bad as quickly. Other questions? So this brings us to the Golden Age of Microbiology. A lot of rapid advances. It's like that with any field. Once you kind of get over that biggest hurdle, these things exist and they do this stuff, you learn a lot about them. So of course it began with Pasteur's work, but after that, many scientists began making connections like the fact that a particular microbe could cause a disease. That we have a whole immune system that provides immunity after an infection. And eventually, we learned about compounds that can inhibit microbial growth without killing the host. Some of the earliest treatments for infections were things like arsenic and mercury, so they work because they kill the bacteria. And you hope they kill the bacteria before they kill you? But Pasteur did a lot of work. He was the person who was responsible for determining that a lot of food spoilage is caused by microbes. He also was the one who determined that fermentation was done by microbes. So you get beer and wine because of microbes. And he was one of the first to link a microbe to an animal disease. Not a person disease, but an animal disease in silkworms. And he also was one of those people who was able to figure out why beer and wine will sour. And it's a little bit counterintuitive, right? Because, like, let's look at wine. Wine is made because grapes get broken down by a microbe. And we talked on Tuesday, and most of us don't want to eat fruits that have been broken down by microbes. But why is wine okay? Well, because it's a specific microbe doing a very specific thing. The yeasts, specifically, ferment the sugars in grape juice to ethanol and carbon dioxide. But what can happen is spoiling of, let's say, again, wine. So wine can be spoiled by other microorganisms, especially what winemakers worry about a lot is bacterial contamination, the acetic acid producing bacteria, because now you're basically talking about there's vinegar in your glass of wine. You do you, but I don't do that. And if your livelihood is making wine, it's very important to prevent those types of bacteria from contaminating your livelihood. So the solution was pasteurization. And we still use this process to this day. It's not designed to sterilize. It's designed to reduce the number of contaminating bacteria as low as possible to prevent things like spoiling. Right? Milk is pasteurized. Can you buy milk at Walmart that has been there for a year and a half? It's not sterile. It will go bad. But you can have it last for a good couple of weeks, especially if you don't open it. Once you open it, all bets are off. But an unopened gallon of milk will eventually spoil. It just takes time.
So like the people who like give their kids like fresh milk, like
Oh, the raw milk?
yeah, is that
Yeah. I don't know. I mean, okay. Yeah. I mean, that's not my thing. I know that's what people did like for, years and years and years before there was pasteurization. And I know people that live on dairies and farms like still drink raw milk. On a scale that we drink milk, like in this country, it would not be safe. And it constantly gets recalled for things like Listeria and Campylobacter. In theory, if it's your own cow and you know that she's healthy and you know that her udders don't have feces on them, knock yourself out. I trust no one. I don't have a cow. So, I was at Sprouts once before I had my kids, and there was a sign. It was like, drink raw milk. It's like the best things, like, compared to breast milk. I'm going, oh my God, what? Please don't give your infant that. And it's like, it has white blood cells. I'm like pus? Yeah, because they get infections in their udders. Like, oh, probiotics. I'm like, and salmonella. Like, as an adult, you make your own choice. And, for the most part, it's probably going to be fine. Not a risk I'm willing to take, just personally. Not a fan of having to run to the bathroom every 5 minutes.
You answered.
Oh, okay. Plus, I don't know. I feel like a lot of the raw milk people, they like to drink it like fresh from the cow. I can only drink like cold milk, so I'm like, ooh, warm milk. That's totally a mental thing. It's a mental block. It's a mental block. Only if it has. I can drink hot milk if it has chocolate in it. You know, hot chocolate is pretty good. You know, like I said, in theory, if the cow is clean, the unpasteurized milk is clean. It will spoil more quickly because it has so many more microorganisms in it. And a lot of people like to use the argument that you lose the probiotics when you pasteurize your milk, and you lose all of the nutrients. So you do lose some of the probiotics because you lose bacteria, but not all of them. So there still are probiotics in pasteurized milk. You do lose, I want to say it's like it's either a B vitamin or a C vitamin, but in the average American diet, everything else you consume would make up for that, like because we don't live on milk alone. Well, I don't know. I felt like my toddlers did for a while, but like adult humans usually don't live on milk alone. So yeah, there definitely are some decreases in those probiotic bacteria, and there are some changes in the vitamin profile. But the other thing to think about is that most pasteurized milk in the US is also fortified with additional vitamins. Like usually A and D are fortified in the milk.
So what makes human milk different for humans?
So there are different proteins in the milk. Like every animal just makes different versions of proteins. Actually, what they do recommend for infants, well, I mean, they recommend formula, but the best non-human animal milk to give an infant is actually goat milk. That they have trouble digesting cow's milk until they get a little bit older. So a breastfed baby or even a formula-fed baby, my pediatrician was like, do not give those kids cow's milk until at least 1 year old, because then they can better digest. They have more acidic pH in their stomach that can tolerate some of the bacteria that are in there. But in terms of like the nutritional composition, there's going to be different levels of fats and proteins in all of the different animal milks, because you know we've all evolved to feed our own children. But yeah, weirdly enough, goat milk, they say, is most similar to human milk. Also not something I tried. But yeah, question.
Do you think we should be drinking milk?
Well, that's a whole, you know. If you are lactose intolerant, no, because I do not want to be in the room with you when you are drinking milk. Evolutionarily, they say, I'm not an evolutionary biologist. The idea that humans now had, by domestication of cattle and the consumption of dairy products, humans had access to nutritionally dense things that had a lot of fat and proteins, and you didn't have to go out and kill something. Like, yeah, you can get fat and protein if you eat the steak, but you have a lot more fat and protein if you can drink the milk for a few years. So there's a thought that perhaps, that allowed for good nutritional development, but yeah, I mean, you start talking about like factory dairy farming and how like unhygienic and disgusting it is. No, we shouldn't be drinking milk.
What do you think?
I give it to my kids, but I don't drink milk. Yeah. My kids like it, but I'm not gonna force them to drink like almond milk. But I still like cheese, so. But yeah, I don't like to eat meat either, because I don't like to eat things I won't kill. I kill plants, no problem, but I won't kill a cow. Not on purpose. It would be a total accident.
I'm not sure how true this is, but I've been hearing that as a society we're growing more and more lactose intolerant because of like a certain enzyme.
It is a mutation. The default is actually to be lactose intolerant as an adult, because you shouldn't be. There's no need for you to be consuming lactose because you are not being breastfed anymore. You're not being nursed. So actually, having lactose tolerance is the mutation. And again, I think that's why some societies were able to, grow, because they had extra calcium, extra protein, extra fats. But But I know when you're lactose intolerant. I can tell. It smells weird. I mean, some people have true milk allergies too. They're allergic to the whey protein in milk. But intolerance is more common. It's not a necessary food product. You'll be just fine without it. But it is for some people, you don't get to go to Whole Foods and buy like $7 bags of apples all the time, right? Milk can be inexpensive and it's a good source of nutrition. So I'm not telling you not to drink milk. Unless you are lactose intolerant, don't do that to yourself. It's very unpleasant. Other questions? But it's usually, again, it's usually safe, and even raw milk, if you get it from somebody who takes good care of their animals, is going to be safe. If you don't know where it's coming from, that's when I would be a little bit more questioning. But so as science progressed, people started to make more connections between animals and microbes. And it was in the 1840s that scientists started to push hand washing on doctors. Again, you're like, legit? Like, you had to convince a doctor to wash his hands? Yeah, you did. It was actually more dangerous to give birth in a hospital than to give birth at home, because doctors didn't wash their hands. And so women would deliver children and then die of an infection. And it turned out that, at the time, doctors weren't necessarily like as specialized. You didn't have an OB and a forensic pathologist. You had a doctor. And they would go from this person to that person and this person to that person. And they didn't wash their hands. It just wasn't a thing. So it was very, very, very dangerous. I think there was the estimates of something like 1 in 4 women would die of an infection if they gave birth in a hospital. And it was much, much lower if they gave birth at home. Still not safe, but safer. And so it wasn't until actually a scientist or a doctor had been doing an autopsy on a patient, and his tech had accidentally stuck himself with one of the metal like scalpels or something, and died of the same infection that killed the pregnant women, that they were like, oh, maybe this guy was right, we should wash our hands. So they finally started washing their hands.
Yeah, I think we're talking about the same doctor, because I saw that this one doctor was trying to get people to wash their hands, and they were like rejecting it, and then they ended up punishing him for that and torturing him, and then he died. And then 100 years later, they were like, oh, maybe he's right. Yeah, it took a long time. I don't know if this was the doctor who had that happen to him, but yeah, there was a lot of it was a point of pride. Like, look at how gross my lab coat is. I've been doing a lot of work. I'm like, yo, that means your lab coat's gross. Like, don't touch me. But yeah, they just didn't know. And then, you have to think about it this way. When somebody tells you, oh, by the way, how a bunch of the women, you delivered their children and they died of an infection, and that was actually probably your fault because you didn't wash your hands? How would that make you feel? So I'm going to be like, it's not the hand washing. It's not, that's not why. A lot of ego involved, but eventually, right, doctors wash their hands now. Those of you going to be doctors, right, you wash your hands all the time. All the time? Yes? You walk out of the bathroom, I'll see you at the sink, right? You always hear the person walk just straight out. You're like, who was that? I want to know. I'm not touching the doorknob. I'm there with my paper towel, like, ugh. Okay. Wash your hands. It's gross. Okay, so this then, when we talk about microbes, leads us to the germ theory of disease. We know that disease-causing microbes have led to the downfall of empires. What happened when the Europeans came to the U.S. and Central and South America? Huh? All the Native Americans, yeah, of smallpox. Yeah. Most of them died of smallpox. And then the rest of them were murdered by the colonists, yeah. It was terrible. Right? It's terrible.
Black Plague.
Yeah, the Black Plague. Oh, definitely. That one's a fun one. When you watch that, you can see the timeline of when it hit Europe. like a third of all Europeans died of the Black Plague? It's a bacterial infection. So we know that infection can literally change the world. Hi, we've been through a pandemic. Nice to see you all. This is a class that was wild to teach during the pandemic. I was teaching microbiology at Fresno State in the spring of 2020, and the students were like, so what do you think about this COVID thing? I'm like, I don't know. I don't know. Don't ask. It's a bad thing. And then, we were at this problem where we knew that humans could get diseases. And sometimes those diseases could spread throughout the population, right? Every person could get the same disease. So, clinicians and scientists knew that there were certain diseases that people would get that only they would get. And a lot of those could be like genetic conditions. But we would also see what we now know of as infections, contagious diseases that would spread through populations. But again, at this time, we didn't know which particular bacteria or, bacteria at all were causing any infection. And so scientists started doing experimentation and eventually came up with things like the germ theory of disease. So Joseph Lister was one of those physicians who participated in this. Listerine, listeria, good and bad. They're going to name stuff after you, whether you like it or not. He developed aseptic technique. They started using chemical antiseptics to prevent surgical wound infection. You needed to have surgery to save your life, and this was, look at the time period, during the US Civil War. So what did they do during the Civil War if a soldier needed surgery? I got this big axe right here. Right? Non-sterile amputations, surgeries, whatever. Lots of infection. So do you take the risk of having the surgery, or you're like, I'm going to live with the thing? Whatever the thing is that I need to have a surgery for, don't care, I'm going to live with it. But when they started using antiseptics to clean, again, they weren't cleaning surgical equipment. Right? Doctor comes at you with a scalpel, or you're at the dentist and you're like, oh man, right? This thing coming at your mouth and you can see other people's plaque and stuff on them. That's the way it was. No evidence, though, that a particular disease was caused by a particular microbe until we got to Robert Koch. He was a German physician, and he was actually the first one who linked a particular microbe to a particular disease. He discovered that a bacterium that we now call Bacillus anthracis causes a disease that we call anthrax. And he did a set of experimental steps, and hopefully you're going, how did he do experiments with anthrax? It's a disease of cattle. Anthrax is typically a disease of cattle. So there were no people harmed during this set of particular experiments. When we talk about the development of the smallpox vaccine, we'll have a different conversation. The development of the smallpox vaccine, the slightly different conversation. In the late 1700s, Edward Jenner, who was an English physician, observed something called the milkmaid's tail. And this was well known. He wasn't the first person to make this connection. But it was well known that milkmaids, who literally were women that milked cows, would get cowpox. Cows get the pox. Cowpox. But milkmaids who had cowpox never, ever, ever got smallpox, which was a big deal in people. And so he thought, what if we take somebody who's never had smallpox, we inject them with cowpox, and then see what happens? And so he did this on a person, the 8-year-old son of his gardener. I don't know what kind of consent was involved in this study. I certainly know that I would not consent to having my child stabbed by my employer and then exposed to a deadly infection. But that's what happened. The boy was inoculated with the cowpox virus, and he was exposed over and over and over again to smallpox, and he never got smallpox. This was not the first way that people protected themselves from smallpox. Prior to this, and I'm talking hundreds, maybe thousands of years prior to this, people were using a process called variolation. And what they did in that process, because they knew if you ever got smallpox, you never got it again. You had some sort of lifelong protection. And so what they would do was take scabs from people who had smallpox. It's going to get grosser. They would take scabs from people who had smallpox. They would let them dry out some, and then they would blow them up the nose of the person who was there for their variolation appointment. The scientific principle behind why that actually worked is that the drying out, if they were dried properly, really weakened the virus. And so when the patient was exposed, they would develop symptoms of smallpox, but because the virus was weakened, they were not severe symptoms. A very mild case of smallpox, but they were always protected in the future. It didn't matter. You got big smallpox, little smallpox. Once you get smallpox, you never get smallpox again. So this was a very well-known procedure. It was well-known in England. Some English noble lady saw it in China and she was like, ooh, we should do that in England. So they did. But there was a risk. What if they weren't dried well enough? You get full-blown smallpox, you're in trouble. So vaccination, which is derived from vacca, which is Latin for cow, cowpox, vaccinia, that's what cowpox virus is called, became the thing. So this was the first vaccine, and that's why they're called vaccines. They're called cow shots, vaccine. They're named after cows. This was it was literally against the law in England to not get this vaccine. And that became very, very important. Smallpox was the first disease that has ever been eliminated from the planet. There has not been a person who got smallpox since 1979. Smallpox does not exist in the wild. In the wild. It exists other places, but not in the wild. We also get to the birth of modern chemotherapy. Often when we think of chemotherapy, or I don't know about you, but when I think of chemotherapy, what I hear, I think of cancer treatment. Chemotherapy is how you treat cancer. Chemotherapy just means using chemicals to treat a disease. It doesn't mean cancer. So antibiotics are technically chemotherapeutic agents. Treatment of disease from chemicals. And so for, again, hundreds, thousands of years, people knew that things like, for example, tree bark extract could protect from malaria. This eventually won a Nobel Prize in 2015. But Paul Ehrlich was the person who coined this term. He was a coworker of Koch that we call the magic bullet. So a magic bullet, the idea behind a magic bullet is this is a treatment that targets the infection without harming the host. Again, they'd give you boatloads of mercury. That isn't great for you. They'd give dyes that they knew could kill pathogens, but it would also kill you. So he developed a synthetic arsenic-based drug. So arsenic-based, you couldn't take a ton of it, but in small enough amounts, it would end up killing the bacteria that causes syphilis without causing damage to the host. So that's the idea. It's a magic bullet. It kills a pathogen, but not the host. And so when we get to like chapter 7 and chapter 20, and we talk more about antibiotics, we will talk a lot more about this, the fortunate accident that led to the discovery of penicillin. So Alexander Fleming was a Scottish physician, and he and his team, I don't like discovered the first antibiotic, maybe the first naturally occurring antibiotic, non-synthetic antibiotic. It was one of the first 3 that was produced, that was not arsenic based, right? And it was an accident because they discovered it because they went on vacation and didn't put their plates away. So they had a bunch of plates out that had staphylococcus on them. And when they came back after their vacation, they realized that on some of the plates there was this mold growth. And in places where the mold had grown, the bacteria did not. There were these areas around the mold that had no bacterial growth. They called it mold juice. We call it penicillin now, which I think is a better name than mold juice. And by the 1940s, this was a drug that had been purified and mass-produced and available for prescription. The 1940s was a difficult time in world history. World War II. A lot of soldiers on our side, because the other side was the bad guys. On our side, they would get things like trench foot, which is a bacterial infection literally in the feet that can cause you to lose your feet. It can kill you. They would get oral infections. During their time off, they would somehow, weirdly enough, end up with things like syphilis or gonorrhea. Don't know how that happened. Penicillin is what got soldiers back on the battlefield. It kept the Allied forces stronger. So what, those Axis people didn't have their own antibiotic? Oh yeah, they did. The first antibiotic, so not arsenic derived, antibiotic, there were sulfonamides. It was a sulfonamide. And it was produced by a company called Bayer. Like aspirin, right? Bayer, pharmaceutical company. Well, Bayer had a couple of things going against it at the time, which made sulfonamides not something that we were interested in looking at. One, Bayer is German, so not great. 2, Bayer also is the producer of Zyklon B. So, there wasn't a whole lot of trust about the things that were coming out of Bayer. Plus, penicillin was better anyway. It was. It is. Penicillin is a more broad spectrum drug. It's less toxic. And we had it, and they didn't. So it really helps when you have soldiers who are healthy and can fight versus ones that have to go home. So the birth of modern chemotherapy really did start with Fleming and the discovery and purification and synthesis of penicillin. But from then it's really exploded, and we do use sulfonamides now. Sulfa drugs, right? They are common. Bayer's okay again. We trust each other. Right? And so when a country is able to make a drug that works, we're happy to take a look at that and use it for our people as well. So the development of antibiotics and synthetic antibiotics has been absolutely crucial in literally making sure that, those in vaccines, that's why we have adults instead of everybody dying as children. If you ever go to the East Coast, you can do one of those like cemetery tours. They're actually really depressing. You'll go to one and you'll be like, hey, this family, this headstone has like 10 names on it, and they were all kids that died within a couple of weeks of each other because they all got diphtheria or tuberculosis and they all died. Like, that's terrible. That's not something we see anymore because of things like antibiotics that can treat infections. So there are a lot of ongoing developments in microbiology. It is a field where, of course, we know that the maggots come from the flies. We're good. Okay, we've learned a lot, but we still have a lot to learn. We estimate that we are only able to culture about 10% of bacteria in the lab. There's a whole big world out there that we don't really know what to do with. We don't know how to work with it. There's a lot that we can study. If you're interested in viruses, well, y'all had a brand new one 6 years ago. Never seen that one before. We saw one that was sort of like it that hasn't come back yet, but where did it go? So virology is really interesting because you get a lot of emerging infectious diseases, things that we've never seen before. Immunology, we learn about how the immune system works. We learn more about it all the time. And then, of course, using computational analyses to look at the DNA. Why are microbes evolving antibiotic resistance? What's happening at the genetic level? How do we know that COVID-19 is different from SARS, which was like 20 years ago? Right? Using modern tools helps us learn about all of those things. Okay, questions about an introduction to microbiology? Okay, cool. We will pick up on Tuesday talking about microbial chemistry. So a brief review of chemistry, but I know you've all taken chemistry and it was your favorite class ever. And you remember everything from chemistry, so it'll be fine. Have a good weekend.